So good afternoon, everyone, and welcome to today's webinar entitled Cancer Variant Prioritization and Reporting Using StrandOmics. My name is Sakshi Shingle. I lead marketing in the Research Informatics division at Strand Life Sciences, and today I'm delighted to be joined by our speakers for the day, Dr. Urvashi Bahadur, Vice President of Clinical Diagnostics at Strand, and Ernie Kim, Manager of Bioinformatics at Celemics. So I thought I'd start off by introducing the companies and setting the context for the webinar today. Strand Life Sciences is a bioinformatics company founded in 2000, whose mission is to support precision medicine across the entire R&D lifecycle, from lab to the clinic to data analysis. To that end, Strand has developed tools, platforms, and services that span these disciplines. Key achievements include a knowledge base for interpretation of solid tumor and heme variants, a CAP-accredited genomics lab that has tested around 10,000 samples in oncology, germline, and rare diseases, a cutting-edge NGS liquid biopsy assay for early detection of oral cancer from saliva, as well as StrandOmics. StrandOmics is a HIPAA-compliant AWS-based platform for somatic prioritization, variant interpretation, and reporting, which has been used to generate clinical reports on the over 10,000 samples tested in our labs. StrandOmics contains information on over 500 genes, 20,000 variants, 750 targeted drugs, which include 104 FDA drugs, 82,000 clinical trials, and 2,300 therapy recommendations, which have AMP-tier classifications. So now Strand has partnered with Celemics to develop a bioinformatics solution tailored to Celemics' assay and user experience. This partnership entails integration of the Celemics bioinformatic pipeline into the StrandOmics environment, providing users with an accelerated implementation, including assay-specific variant filters, to produce high-quality, in-class clinical genomics reporting, and enabling a complete sample-to-answer solution. Celemics is a hybridization NGS-based kit and panel manufacturer, and the only company to do so within the Asia and European regions. Through their proprietary MSSIC technology, Celemics aims to establish itself as a leader in its field by providing innovative solutions to various different fields such as clinical research, synthetic biology, pharmaceuticals, etc. The two main categories, human or non-human, are based on end-user field of study, not sample type. For instance, while samples for pathogen detection kits can come from non-human origins, such as environmental samples, the end users will still be working in human diagnostics. Some human-based panels include the BRCA1-2, OncoRisk and Cancer Screen in Oncology, the G. Mendelione Clinical Exome Sequencing Panel in Inherited Diseases, and in liquid biopsies, Celemics has developed colorectal, breast, and lung cancer panels. As mentioned above, today's panel is regarding cancer variant prioritization and reporting, and so I thought I would walk you through a quick workflow from sample to sign-off of a typical clinical reporting workflow. So you start with primary data generation, which will produce a FASTQ file that will then go through secondary data analysis, to produce a VCF along with demographic information and metadata such as cancer type, location, etc. This data will then go through tertiary analysis. In this instance, it will go to Strand, which will be uploaded in StrandOmics to produce a PDF, which will be reviewed by experts and signed off for delivery. Now for some housekeeping, the chat is live on the side of your screen. You should be able to see it. Please put any questions or comments there, and we will address them at the end. With that, I'd like to hand it off to Ernie to talk about Celemics' target enrichment kit and panel development. Thank you all for listening.
Hello, my name is Ernie. Today I will talk briefly about our panel, especially our pan-cancer 4 or 5 panel. We had an increasing number of customers asking for a comprehensive preset somatic mutation profiling panel. So we have carefully selected 405 genes that bear significance and were most frequently requested for somatic cancer analysis. With this panel, pan-cancer 4 or 5 panel also includes immunotherapy biomarkers, enabling tumor mutation burden and microsatellite instability analysis. And also, when we were creating this panel, we had our competitors in mind, and we ensured that our panel outperformed theirs. So if we look at the next slide, we can see Celemics rebalancing technology in action. A panel can have many regions of interest, but not all of those regions are guaranteed to be captured equally. Some regions are especially easily captured, while others, due to repeats, high GC ratios, or homology, they are really hard to capture. And with Celemics rebalancing technology, we make sure that all regions are uniformly captured. And on the next slide, we can see that using real-life data, Celemics panel shows higher uniformity. Of course, these two panels were designed to capture identical regions and were sequenced using the same amount. On the next slide, we can see that Celemics panel shows superior coverage across depths compared to our competitors. The graph on the right demonstrates how Celemics probes are uniformly capturing even regions with high or low GC ratios. In the next slide, and with superior capture performance of our probes, we can see that even in the very hard to capture areas, Celemics can capture and sequence where our competitors can't. And as we all know, capturing hard to capture areas and all regions uniformly is very important for acquiring confident analysis result. And with confident result, we can generate confident reports using StrandOmics. On the next slide, we have some selected publications in case you want to know more about Celemics' past research projects. And now I'll turn the conversation over to Shakshi, and thank you for listening.
Thanks so much, Ernie. I really appreciate it. I was just informed that my logo and name is backwards. I apologize for that. We flipped them accidentally, and so you're seeing my name backwards. I do apologize for that. Hopefully it's not too confusing. So going back to where we were. So now, thanks Ernie for that introduction. Now that we have our VCS ready and we have our demographic data available, it'll be handed over to Strand, where it can be uploaded to StrandOmics so that our tertiary data analysis can occur, and you can create a PDF, which will then be reviewed and signed off by experts for delivery. I will hand it off now to Urvashi to explain that aspect of everything.
Thank you, Sakshi. So, as Sakshi and Ernie have explained, the typical workflow is that a sample goes through the lab on an NGS panel, and in this case we've seen Celemics panels and what they're like. Then it goes through secondary data analysis, and the output of the secondary data analysis is a VCF, which is nothing but a long list of variants of various types. And now what is really important as an end user, or what is required for a clinician, is tertiary data analysis needs to be performed, which is from this list of variants, the clinically significant variants which are important in cancer and which are likely to have therapeutic implications, these need to be determined, and then a report needs to be created which can be quickly reviewed and signed off. So this is the process that we do at Strand, and we can take VCFs which are generated from samples run anywhere in the world, and then quickly create a report and hand it over to you. Now, the software which allows us to do this is StrandOmics, and this software is composed of a proprietary curated knowledge base as well as various rule sets which allow us to do this in an efficient manner. Now, as a customer, the first thing you need to do is input the VCF and some other data. So in the next few slides, I'll show you what that process looks like. The first thing that needs to be done is the sample details need to be entered, the cancer type and anything else that is required, and of course the data. So in the next slide, let us look at a snapshot of what the view looks like in StrandOmics for this. So you can see two activities here: register test request and data upload. Register test request is nothing but when you are creating an entry for a sample, and it's a quick process where you will actually put in the various things that are needed, such as the cancer type, the patient name and age if applicable, anything else that you require to be shown onto the clinical report. So in the next slide, let's look at what that looks like here. So this is just a quick screenshot of some of the fields, and there are many more fields that may or may not be entered, depending. A few of them are, of course, essential, and then the rest you can add if need be, if that's what you need. So the basic information, basically the cancer type and a few things like that, need to be entered. In the next slide, we can look at what happens after this is done. So we've registered the test, the sample. Now we need to associate the data with the sample. And there are 3 easy steps for this. So, you'll be prompted on the data upload screen to attach the get the variant file, which is a VCF in a standard format that we accept. Then you upload it, and then you sign it off. So once you sign this off, this is done, and now the data is available to us for actually looking at it in StrandOmics. The clinical team, which is the team that I have, looks at the data and actually understands what needs to be done for creation of the report. So now I will show you a demo for this and go over this. I should add that if there are multiple bulk samples, then we can, of course, do this in a programmatic way as well. So once we have this VCF, what you see here is now on my screen, I hope you can see this, is a whole list of variants on this case here. Now, once you have this list of variants, what you can see is that each one of them also comes with a little bit of a label here, if you notice on the right. And here, in this case, we have 300 variants for this case here. And so, depending on the size of the VCF and the panel, etc., this number will vary. What StrandOmics does is it quickly sorts through this list so that you don't have to go through all 304. It automatically assigns them a priority, if you will, based on how important these are likely to be. And you have to just quickly do a sanity check, look at everything, and then take these variants into the report. So if you can see, the first few variants here are saying gain of function, loss of function, etc. But then later on, we have variants which are saying cosmic and things like that, and then even lower, and then you have diminishing in here. So this already sorts the variants in order in which you need to look at it. Now, we also have details on each variant, but before that, let me also show you that we have a couple of tabs here. If you notice, this is the investigational tab, but we also have another tab called standard of care. Now, in this case, the case that I'm showing you today is a colorectal cancer case. So the standard of care tab actually tells you about those variants which are very important for standard of care drugs for this cancer type. Now, as you would know, in certain cancers, there are indeed, genomic findings which are well known enough and well established enough to guide recommendations for standard of care therapy, and these are in clinical practice guidelines and sometimes on the FDA label of those drugs as well, that you need to test for certain markers before a patient is eligible for that drug. Now, this is not in every cancer, but for all the cancers where this is the case, we have a standard of care tab as well. So in this case, in colon, you have KRAS, which has shown up. So there are many genes in colon which have this, for instance, KRAS, NRAS, PRAS. But in this particular case, what we've seen is that a KRAS variant has been identified and a PIK3CA variant has been identified. And so these are the two variants which will need to be looked at from if you're only interested in standard of care drugs, for instance. And then you're back to the investigational tab, which shows you all the other variants which will have implications, not just may or may not have for standard of care drugs, like these two have for the standard of care drug, but also other variants which can have implications for drugs in trials. So once we have that list here, we can go through each of these variants in detail. Now let's look at what a typical variant card has. So once we look at a variant, sorry about that. Yeah. So once we look at the variant here, now let's look at, say, something like MIC, which is a copy number variant. So when you open this variant card here, you're going to actually take a look at what is actually written about this variant. So this is an amplification variant. It has been reported previously in certain cases, and this tells you why it's important. What is the reason for it, that it's actually associated with an overexpression, And it also tells you a little bit more about the weighted score, etc. So once you know this, and you can say yes on this, as you can see, there's a decision for each of these variants. And once you have this decision, you can go over. Now here, you can only see the copy number variants because that's what I had shown you here. But now you can apply filters. You can also see the point mutations, or you can see all. And you can also, for instance, look at something like the supporting read. And once you apply that, you can see that something only, so here you're back to the rest of the variants, not the copy number variants, and then you can take a look at these as well. So once you have this, now you can choose each variant that you want, and you can quickly take a look at them. And now you go to selected variants, quickly take a look at them in the report, and you're going to see. So in this case, the variants that we have, we've chosen 4. There's CDK4, KRAS, MYC, and PIK3CA. So let's see that once we know these are the gain of function variants, what we will do is now look at the therapy implications for each one of them. So now that we know that we're supposed to look at the therapy, let's look at what that looks like. What we have here is that quickly look at the drugs for them. Now, in this case, you have abemaciclib, palbociclib, ribociclib. We've chosen to look at the CDK4 matrix, which is what we call it. You see here, this is colorectal cancer, and we have this for each particular cancer as required. So different cancers will have different drugs and different evidences. As you can see, these drugs all have markings here. So you have an FDA-approved drug, if it is indeed FDA-approved. If it's a drug in trial, it will have only a drug in trial. You have a summary here, which tells you why CDK4 is important. And then you have also the clinical trials. So we access clinical trials from the NCI's clinical trials database. And when we do that, we get all these various details about the trial, the title, the ID, the locations, the phase, everything. So this is available to us. And this helps us in making a decision on whether actually this drug is under an active area of study or it's not. So once we decide that, yes, these are the drugs that we could go ahead with over here, then we can simply go to a report. And this is what a report looks like. So we can go to this view here now. And when you look at the report, so if you notice now, here is the demographics section. All the demographics that I talked to you about in the first view, those are the ones that are here. And if you notice here, these are all redacted. So StrandOmics is actually HIPAA-compliant and role-based access. So in this case, if you enter certain things which are not required for our team to see, except the cancer type and the cancer other details, then we are not going to really be seeing these. And so it's only the person in sign off and at your end who can take a look at all these data. So that makes it HIPAA compliant. So here, these are the things we entered here. Now, then we go on to the clinical indication, and then we tell you what are the variants that were detected. Now, in this case, we have 4 variants detected, as I had shown you in the previous slide. So we have PIK3CA, KRAS, CDK, and MYC. And once you have these, you can now take a look at the tier classification as well. Our report is compliant with tiers. So what that means is you have a tier 1 and a tier 2 for the important variants, and tier 3 are VUS, and tier 4 is likely benign. So tier 1 variants are those which have a very strong clinical implication in this cancer type, and tier 2 are those which are well known but not so strong yet. Now, coming to the report itself, you have a section for standard drugs, and this is the section that we looked at, if you remember, in the standard of care tab. And here what you can see is that here you have the 2 drugs which are approved in colon cancer, which is cetuximab and panitumumab. And what you have here is a poor response because you have some variants here in these genes. And now moving on to the other section, which is really about other novel investigational options that can be available. So you have all these over here, and you have CDK4, KRAS, MYC, and PIK3CA drugs, which are there for each one of them. Here you have the approved indications for some of them. You have clinical trials where the clinical trials are there. If the drug is not yet approved, you get a none. So you know that this drug is still only in clinical trials, whereas these drugs are approved in other cancers, but not in colon cancer. So this gives you, the first couple of pages give you a quick snapshot of what is really important in this case. Now, moving on to the Other sections of the report, the details. So here you have all the details. So you have the drug or response. This is the evidence which tells you about the trials, or the data which tells you, what was the survival or anything like that. So you have this for both these mutations here. And then you have the details of the variant. So this is actually what they're telling you about the variant. So we saw these variants earlier. Now this is really the description that we have, and we have description for, we've looked at over close to 20,000 variants and seen which ones of them actually have a gain of function or a loss of function associated with it. And so that's what we have here. So that's good. It should be noted that here you have two variants for the same drug, and we do have a concept of a combinatorial response also, because in certain cases you have something like, say, T790M and MET or something like that in a lung case. So you can actually, have two variants impact the response to one drug. So similarly, we have the same for cetuximab, and then we go on to the investigational section here, which is the abemaciclib, for example, the drug we saw earlier. And what we saw here is that the evidence is good for CDK4 here, and it tells you about clinical evidence. It also tells you about preclinical evidence. Sometimes the evidence may not be from the very same drug, but from other drugs in the same class. So each drug has also, we have the concept of a drug and a drug class, and all the drugs are mapped to it. So, you can tell, so evidence from related drug is also sometimes shown for that very clearly. And then you have the marker details, the clinical trials now, the ones which you saw on the first page. And then this tells you which ones are actually important. And like some of these actually are marker-driven clinical trials, and some might be general clinical trials, maybe in the same cancer, or it may be in another cancer type, or just overall in solid tumors as well. So we have these links here. Our sources are clinicaltrials.gov, IRTRP, and WHO and NCI clinical trials. So once you go through the report like this, the references are below here. All these are hyperlinked for you to take a look at. And then, so this is quickly what happens, and this is what the interpreter looks at these and generates a report. Now, this report is available for you for sign out, or we can at Strand also sign this out ourselves. And if you just go through towards the end here, I'll show you one for one of the markers for prognosis as well, where sometimes you may not have a therapy recommendation, but you may have something which is important overall for the course of the disease. So we have a way to put that in. And after you have all that, we have you can have signatures, which can be from your end. You have supplementary information if you want to add something from your side about how it's done in the lab or anything like that. So those can be added on as well. The header and footer can also be customized, or we can have Strand header and footer. And so in this way, we can take any VCF and quickly turn out a clinical report in some time and send it back to you, which you can review and take a look at it, and that's for you to deliver to your patient. So I hope that gives you an idea of what we do here. And with that, I'll give it back to Sakshi.
Thanks so much, Urvashi. So with that, we finished with the presentation side of the webinar. I hope all of you enjoyed it. So we do have 2 questions at the moment, and if there are any more, please feel free either on the YouTube or on the LinkedIn to send any of your questions. So the first one is from Violetta V. I hope I'm pronouncing your name correctly. She asks, what is the annotator used to annotate the variants coming from the VCF file? If either you or Rishi or Ernie could take that.
Yeah, no, if the question is, can be explained a little bit more, but I'll answer what I think. So we annotate the variants, with data that we've generated got from literature and multiple things like that. Maybe the question can be elaborated a bit, then you can answer it.
Okay, so we can wait for her to say that. While we wait for her, maybe Violetta, if you could clarify your question a bit, that would be great. In the meantime, Christine Yan asks, how do you ascertain or verify that the variant identified in the VCF is a true observation and not a sequencing error of bioinformatics analysis or error bias?
Right. So I can explain what we want, and then I don't know if Ernie wants to answer something as well. So it depends on the secondary pipeline, of course. But after that, what we have is we can take a look. So typically, what we've seen is that if there's some variants which come very frequently, these kind of errors or systematic errors, these are panel artifacts. So what you can see actually in StrandOmics is it tells you how many times this variant has been seen before in the same panel or in other panels, and that gives you an idea whether it's a noisy location or whether it's a panel specific artifact. You can also look at the neighboring sequence and see whether there's for instance a polymer region, and so those allow us to do that. That said, we ask our customers to also provide whatever cutoffs they want. So if you notice, I put in a cutoff of 5%, and so if somebody is not sure, which is 5% is a typically accepted cutoff. So we wouldn't really go for variants below that and that sort of thing. So as a customer, you can tell us what are the cutoffs that you want, and for things like something like a strand bias and those things as well. So I think maybe Ernie wants to add to that.
Well, it was very nicely explained. So I want to first say that you cannot be really 100% sure whether a variant is true positive or a false positive, because anything can happen. So what really matters is how someone, our customers, may set their cutoffs. They might decide that I'm not going to consider this as a variant, but it's really a hard, tough process. But once you get used to a panel or analysis programs, you kind of get the hang of it. And there's some situations where if you look at one variant specifically, you can tell that why this variant is there. So like, as Urvashi mentioned, it can be due to strand bias. And if we look deep into it, we know why strand bias is happening here and consider that variant as a false positive. Or it may be due to some problem with the homologs. Sometimes those kind of things can make false positives. So it really depends on what kind of variant we're talking about, which position and which program and which panel. So it's really a hard question to answer.
Fair enough. Thanks so much, Urvashi. Thanks so much, Ernie. So our next question, we actually have two from Sudipta Sengupta. Again, I hope I pronounced your name correctly. I apologize if there are any mistakes. So his first question is, do you practice pathway analysis for preparing the reports into the backhand? How could you recommend a drug for multiple usages?
Sorry, do you perform pathway analysis too? Could you repeat that, Sakshi?
Yeah, so he says, do you practice pathway analysis for preparing the reports into the backhand? And how do you recommend a drug for multiple usages?
Okay, so pathway analysis, yes. So the screen that I showed you where there was a list of drugs, right? And for CDK4, you just magically got abemaciclib and palbociclib and those things, which are actually CDK4/6 inhibitors. Now, what happens is that, yes, we do know that in this case it's a direct target, but sometimes it may not be an exactly direct target. But even so let's say if it's a RAF, but there's a MEK inhibitor or something. So yes, we do have the concept of a pathway. And we do, so we curate drugs with their target, and then we curate them as upstream or downstream of any panel gene. And in that sense, take into account the fact that whether they will be able to inhibit something where a mutation is found, a gene that's upstream. So yes, that is taken into account. And exactly, so there's been a lot of background curation for this. And so the list of drugs that appeared for CDK4 will be different for than the list of drugs which appeared, say, for KRAS. So that in the back end, we've exactly used the pathway analysis and the literature evidence to come up with which drugs should show up for which gene. So yes, we have that. And then, was there a second part?
So there's a second part to the first question, where they asked, how can you recommend a drug for multiple usages?
we recommend the drug for different usages based on the clinical trials and the clinical evidence. So sometimes one drug, maybe if you have 2-3 genes and they all happen to be in the same pathway, then you do recommend those as well. But those are again in investigational or in trial. So we don't recommend a drug if it's not in trials and there's no evidence for it. Just because of pathway, we wouldn't do that. We would go and look at evidence for that drug or for that class of drugs and then only recommend it.
Got it. Thanks so much, Urvashi. So their next question is, do you also follow the VITAL tool designed by CAP during variant interpretation?
We don't need to use something specific like that. We've incorporated multiple things in StrandOmics itself for us to understand whether the variant is gain of function or not. So we just use, we use Cosmic, we use databases, germline databases to find out the allele frequency. We use bioinformatic predictors. So we use a combination of all these, and then that's how we look at it and understand that the variant is important.
Got it. Thanks so much. So Violetta did respond. She said that her question is, what is the software used as an annotator to annotate or describe the variants that have been filtered as clinically significant? Some examples she gave were ANNOVAR, variant effect predictor, or so on and so forth. Does that help a little bit?
Right. So I guess, so StrandOmics does that. I mean, I think we've built our own. So pretty much StrandOmics is the software which takes into account these, various databases plus curated knowledge base and then assigns this label. So it's, it's our own software that we use, and that's what StrandOmics does. So it's not just a software for creating a report. It's also a database which incorporates data from other databases and rule-based sets. So if you have something which is a loss of function, if you're a truncation mutant, it tells you all that. So it does multiple things, and that's how we come up with the final. So you can actually see that yes, this is a frame shift at this position, and this is what's happening. So yeah, we do all that, take into account all in StrandOmics.
Got it. Thanks so much, Urvashi. So I think that's it. We don't seem to have any more questions at the moment. I can wait maybe a few seconds, but otherwise I think that's it. Yeah, that sounds good. Thanks so much, Urvashi and Ernie, for joining our webinar and being such gracious speakers. We really enjoyed, at least I really enjoyed your talk, and I hope everyone else did too. And yeah, I think with that, maybe if you do have anyone has any inquiries, please feel free to reach out to us on these emails. For Strand, it's sakshi.divija@strandlifesciences.com. For Celemics, it's support@celemics.com. And yeah, I think we can end there. Thanks so much again, everyone, for joining. Thank you, Urvashi and Ernie. I hope all of you have great